Welcome to episode 24 of Travelling Science. G'day legends, welcome back to Travelling Science, the podcast that's sharing science with the world. My name's Crowe and I'm the Travelling Scientist, coming to you today from my home in Aireys Inlet, Australia. I'm a health science communicator, and on this podcast we interview doctors, researchers and experts, hearing their stories and sharing their ideas that can help us to live smarter, happier and healthier lives. This week's guest is Dr. Taylor Szyszka, a protein engineer and synthetic biologist who has spent her career exploring the fascinating world of proteins and how we can use them to build sustainable systems across different platforms. Dr. Taylor is also passionate about STEM education and engagement, and she's been involved in numerous science communication endeavours, from TV to radio, game development, and even stand-up comedy. And it shows. She's super witty and somehow manages to make her science both informative and hilarious. And during our discussion today, Taylor answers questions including what is synthetic biology, why plants suck at photosynthesis and what we can do about it, how to share science through comedy, how does one engineer proteins, and how empathy drives scientific discovery. And we also discuss things like engineering microbes, nano-reactors, carboxysomes, encapsulins, the structure of proteins, bioremediation, a new science card game, and even crocheting. It's just an awesome conversation, and I'm sure you're going to enjoy it. So let's get right into it, talking synthetic biology and science comedy with the one, the only, Dr. Taylor Szyszka. Dr. Taylor Szyszka, welcome to Travelling Science.
Thank you so much for having me.
No, it's an absolute pleasure. Thank you so much for taking the time to come on the show today and have a chat with us. Now, my very first question is, can you explain what is synthetic biology?
Yeah, that's a great question. I think sort of the simplest way to describe it is it's applying principles of engineering to biology and biological systems. So we're talking things like DNA, proteins, or microorganisms, things like that. And so basically it's engineering them to serve a different purpose or work in slightly a different way to sort of address other problems.
That sounds very cool. And so how do we do that, or why do we do that to improve our world? Like, what are the potential possibilities?
Yeah, that's another great question. I think I'll talk about some of the applications that are kind of like out there in the world at the moment. So there's folks working on things like engineering microbes to produce alternative proteins for things like plant-based meats or milk alternatives. There are also people working on things to make dyes or textiles more sustainable. A lot of these synthetic biology, let's sort of end applications, are around sustainability or doing things in sort of a cleaner, greener way using biology and microorganisms. There's also really neat things where some people have sort of engineered enzymes that can break down plastic to sort of address plastic pollution or infinite recycling and things like that. So there's a ton of applications, and a lot of them are in this idea of doing things sustainably and more greenly. And biology lends itself really well to that.
Absolutely.
And so a lot of it, because I guess synthetic biology is pretty young on the scale of fields. Like if you think about biology and chemistry, which have been around for hundreds and hundreds of years, synthetic biology is about 20 or heading towards 25 years old, so it's pretty young.
Synthetic biology is younger than me. Not even 25 years old, and it's already helped to greatly improve the fields of medicine, biotechnology, agriculture, and sustainability. Well, I'm over 30 here, and I struggle to put out a weekly podcast about this stuff. So how do we use synthetic biology?
It's sort of using these ideas that we've learned over hundreds of years or even millennia of studying biology in the natural world, and now we can use that information that we know and those facts that we know for different applications.
Nice. Yeah, it sounds very futuristic, almost like an oxymoron, like biology is the study of nature and synthetic is human-made. But the potential is really cool, like the fact that we can change natural microbes that are already doing these processes. If we can change the processes slightly to benefit the world in some way, and to improve sustainability in ways like breaking down plastics that are being built up, or producing fuels, or whatever else they can. I forgot what else they can do, but those things.
They could do lots of things. There's also like bacteria that can grow on like carbon dioxide or methane or greenhouse gases that you can then sort of engineer to produce things like bioplastics or to become like feedstocks for livestock. So there's a lot of neat applications, and basically just nerding out and listing all of the cool things that I've learned while working in this field.
We hear about these things quite a lot, like the potential for microbes to break down all the plastics and the potential for microbes to fix the bad gases in the air and improve climate change. I feel like there's a lot of potential, but is it actually happening, or is it like taking time because of the research, or is it just some scientists sitting in a lab saying like, wouldn't it be cool if we could just do that? But is it happening, or where are we at with implementing these things?
Yeah, so a lot of the things that I've listed already are startups and companies that exist and are doing these things now.
Probably at like a small scale, and it must take time to build up, I imagine.
Yeah. Because I think that's one of the things that I think folks can get kind of spooked about when talking about cool synthetic biology things and applications and stuff. They're like, oh God, it's happening right now. And the work towards it is happening right now. But there's a lot of steps between now and that amazing bio-based world that we're trying to build. So there are like, and I work in an academic lab, so I'm sort of at like the beginning part where we're talking about wouldn't it be cool if, and doing the proof of concept stuff to try to make these applications happen. And then that turns into sort of industrial translation, commercialization, and then that takes a while to be able to produce things at the right scale, and to get enough financial support and sort of make sure you get all the regulations crossed off and everything. And so it is a long process, but there are a lot of exciting innovations happening at every single step. So we are getting closer to sort of realizing a lot of these exciting end goals.
Fantastic. And I know that's really how it starts, I suppose. People sort of sitting around being like, wouldn't it be cool if we could develop a microbe to do this? And then you go and do it. And it might take 20 years, less or more, who knows, but yeah, it all starts from an idea, so it sounds super exciting. You mentioned your research. Would you like to tell us what you're currently working on, or what you've been doing lately? Like, where do you fit into synthetic biology?
Yeah, definitely. So I've spent up until now talking about microbes, and that's where I'm going to stop talking about microbes, because I don't work on microorganisms. I don't work on organisms. I have so much respect for people who do, because they're so complicated. So I work on proteins. I'm a protein engineer is probably the most accurate to what I do every day. So I specifically work on protein cages. So there are these little sort of, I guess, hollow spheres that are made out of protein. And the ones I work on are about 40 nanometers in diameter, so it's about 1/100,000th of the thickness of your fingernail for each one. So they're very, very tiny.
The thickness?
Thickness of your fingernail.
Oh my God, that's very nano. That's incredibly tiny.
Yeah, it is. It is extremely nano.
These protein cages are so tiny that if you lined up 100,000 of them in a straight line, they wouldn't even span the thickness of your fingernail. That's less than 1 millimeter. 100,000 of them. This is some serious nanotechnology right here. Right. Okay, so very, very small spheres.
Very small spheres. And the one I work on the most is, it's called encapsulin. And the particular encapsulin that I use is usually storing iron in like cool little bacteria. So encapsulins are a family of bacterial proteins that bind together to form these nano-sized compartments or cages in order to store things inside bacteria like iron or other essential nutrients. And the idea here is that we could use those encapsulins. Actually, I'll let the protein engineer explain this. The cool thing about them is they actually self-assemble on their own, so I don't have to do anything to make them come together and form this sphere. It's like 240 copies of one protein that has stuck to many, many copies of itself to form this amazing, perfect little sphere. They're really incredible. And we've figured out ways that we can put other proteins and enzymes inside of them. And besides having protein-covered protein with protein filling, which is definitely what a protein engineer wants, we are interested in using them to make nanoreactors.
So nanoreactors are essentially teeny tiny little factories that can be designed to create certain products. Like imagine you had a bunch of soda streams, but instead of pushing a button and getting some soda, they made a specific enzyme or protein that you wanted for some reason. Now shrink them down to 1/100,000th of the size of your fingernail, and you've got nanoreactors, which can be used to make almost anything at a nanoscale.
So doing interesting chemistry with enzymes on a really small scale in like a confined space. So there are potential applications of things like biomanufacturing, which is a field I've learned about since sort of starting to work in synthetic biology. But having enzymes make stuff for you, which is amazing because they're just really incredible little chemists. And then the other major project that I'm working on is trying to improve plant production by improving the way plants do photosynthesis. So I was very surprised when I started this work that plants are actually not amazing at doing photosynthesis, even though it's how they produce their own food and extremely vital for them to be able to grow.
Who knew that plants suck at photosynthesis? Apparently they only convert between 1 and 5% of the sunlight into energy. Pathetic, right? Well, Dr. Taylor, along with many, many other scientists, are looking at ways we might help our poor little plant pals. So what is the problem here? Why do plants suck at photosynthesis?
But there's a protein involved in photosynthesis that's really slow and really inefficient, and that creates a bottleneck in plant growth. And so every organism that does photosynthesis has to deal with this problem. And algae have a really cool solution, which is this thing called a carboxysome, where it basically traps this naughty little protein inside of another sort of hollow sphere, but it's perfectly engineered by nature for it to work more optimally. So it's faster, it's more specific, it doesn't have the same problems that you normally see, and so then algae get a benefit from that. And so people have been really interested for decades, okay, how do we put this carboxysome into plants and give them the same benefit? Then they could grow more efficiently, they can get better photosynthesis. But the issue with carboxysomes is they're super complicated. They have that protein cage structure still, like the same sort of idea, but it has a bunch of different parts, and it just doesn't reproduce in plants as cleanly. And so I'm trying to use my encapsulin, my protein cages, to make a more nano version of that that's compatible with plants to try to improve that photosynthesis and try to improve plant growth.
Very cool. So you're doing all the things with encapsulin.
Yes, all the things with protein cages, basically.
You said you were trying to potentially build nanoreactors to do what was that word?
Biomanufacturing.
Biomanufacturing. Can you talk about what that is? Just briefly, like what sort of things would we be biomanufacturing? Like what can we create with these tiny little chemists, these nanoreactors? It sounds cool.
Yeah, I mean, it's an incredible name. That's already there. And biomanufacturing is like this really neat, very young field. But basically, like, if you're trying to do chemistry, as a non-chemist, I find it daunting. And the syntheses and like the steps you have to go through to produce like different small molecules for different, purposes, whether it's like therapeutics or like sensors or anything else, they can be really laborious, they can be really complicated, and then they can also be, you can have some industrial issues down the line. Enzymes are incredible chemists. They've, nature has been optimizing their activity for eons and eons, and so they're just so good at what they do. And if we can find ways to sort of optimize their performance through giving them a little space where they can work better and try to capitalize on those gains, I guess, that's even better. So I guess for us, we're really interested in what the reality is of doing chemistry in a femtoliter space.
What is the femtoliter space, you ask? Well, it kind of sounds like it could be a remote part of our ever-expanding solar system, Or it could be a weird new nightclub that's just opened up and already has a reputation for sticky floors and seedy bartenders. But no, femtoliter is actually several orders of magnitude below nanoliter. So you've got liters, then milliliters, and smaller than that is microliters, and then nanoliters, then picometers, and then femtoliters. That's 10 to the negative 15 liters, aka a really, 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 really small amount of space.
So basically, this teeny tiny little nanosphere, like if you smush a bunch of enzymes into a small space, they probably won't be happy. Much like myself, if I'm shoved into a small space with a lot of other people, it's kind of hard to focus.
Nobody likes that, yeah.
Yeah, exactly. It's the same for enzymes. They're not that different from us. They're just much better at chemistry, at least than myself. I don't want to have any chemists coming after me. Definitely better chemists than me. But yeah, and so we have to think about things like how do we control how many enzymes are getting packaged? So there are different sort of little engineering tricks we can do to control that. Or, for instance, a really big complicated enzyme that maybe has a lot of parts that need to come together. How do we package that whole thing instead of just half of it, which can sometimes be a bit of a problem because everything's happening in the cell at the same time. Like, are things being packaged or going into the shell as the shell is forming? It's all very chaotic. And so finding different ways to control these processes. So it's really kind of like designing a little factory, sort of, but just in chemical space rather than like physical space.
True. And the fact that this is all happening at the nano and femto scale is just, it's mind-boggling. And how you even figure these things out is beyond me. But let's not get too much into that today. Let me talk a little bit more about the photosynthesis project you're working on. What happens, you mentioned before these carboxysomes, what happens when you try to put those in a plant? They're not happy?
Yeah, so a lot of the issue has to do with that shell protein. Because it's a cage the same way that our encapsulants are a cage, but they're just much bigger and much more complicated. So they have a few different parts. And so when you try to produce that in plants instead of in algae, it can be really hard to make that whole shell.
Tricky.
And the shell is complicated because it does a lot of amazing things, like what I was talking about with pores. People think it's still kind of contentious, but people think that they sort of keep CO2 in and then don't allow oxygen in. And when you're dealing with things that are that small, that's really, really impressive that there's any kind of barrier there that can be selective. So yeah, the shell is incredible, but it's also complicated. Whereas our shells are very simple and we're engineering them to make them incredible.
Love that. Love an incredible shell. And I guess the potential here is, as you were saying, if we can use this concept to improve photosynthesis, the potential is to improve plant growth, improve crop yields, make more food for a growing population. Is that kind of the idea?
Yeah, that's kind of the big, like, dream end goal that we have in the back of our heads when we're working on the really nitty-gritty details. And it's like, oh, like one day this could actually be really, really powerful. I guess the other side of that same coin of, improving photosynthesis, the step of photosynthesis that we would be improving is called carbon fixation. So that's when carbon dioxide sort of goes into the plant and then gets converted into sugar. And so that has a lot of really far-reaching applications as well, of just being able to utilize carbon dioxide in the atmosphere better for biological reasons.
So many reasons. Awesome. I love that. Can you just talk about your goals a little bit more? You said that the overarching goals. What do you hope to achieve with your synthetic biology visions? Where are we looking?
I mean, I guess, like, speaking for myself and sort of the very niche area that I work in, I'm really excited at the prospect of all the amazing things that proteins can do. And I just really enjoy and find fascinating the fact that these protein shells, these encapsulants that I'm working on, store iron in bacteria that live in like hot springs. And now we're trying to use them to improve crop yields in plants. And I think that's the really amazing part of synthetic biology, is you can take something that is totally unrelated and find a way to engineer it to have like a new life and a different application. I think that's really powerful, and like this is just one small example, but synthetic biologists all over the world are taking ideas like this, taking something that does one thing changing it a little bit to do something else that can really transform the world that we live in and provide these amazing, transformative, sustainable technologies. And so those are the things that get me really, really excited.
Yeah, you just smush some proteins together and you can be doing something incredible.
Yeah, we can manufacture stuff. We can improve photosynthesis. Yeah, there's so many applications that we're really, really excited about being able to achieve. Especially with just these like funky little shells. They're so cool. They're really pretty. Like, I'm a big fan.
Can you like look at them under a microscope? How do you visualize these nano-sized structures?
Yeah, so we can use an electron microscope. It's like a light microscope that you would use in school or something, but instead of light, it's electrons, so it shoots an electron beam. So it allows you to see stuff on much higher magnification. But yeah, we can look at them, we can see little circles. It's really cool, because there's not a lot of proteins that you can just see, so yeah, it's a nice little check. And we have other techniques as well that we can use to see them indirectly. But one of the realities of being a protein engineer is I spend most of my day transferring drops of clear liquid from one tube to another. So it can be hard to remind yourself that actually there's tons of little shells in here. And it just looks like water going from one tube to another.
Yeah. I'm doing groundbreaking, world-changing work in this tiny little tube here, and you can't even see it because it just looks like that little bit of liquid there. Most of the stuff I studied at university, microbiology, immunology, virology, bacteriology, parasitology, all these things, you couldn't see them. It was all just in books. And so I was like, I'm pretty sure what I'm doing is real. I think it's real.
Yeah, I'm pretty sure it's cool. We've had people who have like wanted to film stuff in the lab, and they'll come in and be like, oh, can you show us something really cool? And then I'm just like, well, here's all of my proteins. Yeah, they don't look like much, but they're really cool. Until you start using fluorescent proteins. Fluorescent proteins are very cool.
I talked to Crowe, the scientist, last week, and she was like, I started working with GFP, and I just got super into microscopy because it looked cool, which it totally does.
Yeah, I definitely have moments where I'll like engineer a different like aspect of the shell, and I'm like, look, I just want to make sure it works. I want to be able to visually track it. I'm going to put a pink protein in it. And like, yes, there is a scientific reason for it, but there's also like a personal, emotional reason for it, which is that sometimes I just need a fluorescent protein in my life.
I'm going to make it light up. This makes me want to talk about your comedy stand-up, which you did, science stand-up, which you did recently. How long ago was that?
That was in August and September of this year. So just a few months ago.
That was really cool.
Thank you.
It was super informative and super hilarious.
So in the show notes, there is a link. And if you click on said link, you'll be transported to a world of wit and wisdom, aka Taylor's science stand-up comedy, where she explains her research in a relatable way by comparing it to the new Barbie movie. So go, watch, and simultaneously laugh and learn science. Because
I thought it was an awesome way to explain what you're doing, and it was hilarious. How did you get into? Did someone approach you about doing a comedy routine for your science, or how did that come about?
Yeah, so I do a lot of outreach because I really like it, and I've kind of done outreach as long as I've done science, and I can't really do one without the other. I've noticed I'm not really happy if I'm doing one without the other.
I need to share this.
Yeah, they go together. And I was really fortunate this year to be part of the Superstars of STEM program run by Science Technology Australia, which is all about raising the public profiles of women and non-binary scientists. So it's an amazing, amazing program.
Superstars of STEM is a program run by STA, Science and Technology Australia, and it aims to smash society's gender assumptions about scientists and increase the public visibility of women and non-binary people in STEM. Check out the website if you want to find a bunch of scientists doing cutting-edge research and sharing it with the world, including a few that I've interviewed on this very podcast, like Dr. Lily Van Eden, episode 20, where we spoke about psychology and nature conservation, and Dr. Amanda Curry, up and coming, I want to say episode 27, where we get deep into the environmental and ethical aspects of epigenetics. But what does Superstars of STEM have to do with science stand-up comedy?
And And a couple of other scientists had done this comedy program in May where they sort of did their pilot program. And they talked super highly of it. And so then I saw like an EOI go out for something during like July, August, and I got really excited. So I signed up, and then we did like a month or so of workshops, working with a comedian and with a speechwriter, like communication expert, on how to inject comedy into a 10-minute science talk.
And you did it so well.
Thank you. Thank you. Yeah, it was so fun to put together.
Yeah, no, I nearly did something similar. There was something this time last year. It might have been the one you were talking about in May, actually, because about this time last year I was getting involved in it. And I sent them a pitch, and they said, "Oh, it sounds great. We'd love to get you involved. You can come. You can work with Dr. Carl. We'll walk you through the science, and we'll get you ready to do your stand-up bit." But the timing didn't work out because, one, it was in Sydney, and two, I went to Japan. Japan was awesome.
That's amazing. Yeah.
Yeah, but I didn't get to do my science comedy, and I've seen yours, and it's so good. So if I ever get that opportunity again, I would love to do that.
And so this one's actually going nationwide. So if you're interested, if any scientists are listening and very interested, I can highly recommend it. And so check out Future Science Talks, and they've got like EOIs and stuff on their website for the comedy program. But yeah, they're going to be all over the country, which is amazing. As many scientists as we can turn into comedians as possible is a good thing for outreach and communication.
It's such a good way for science communication. I feel like if you want to go to a science lecture, no, but do you want to go to a science comedian where you can laugh your ass off but walk away being like, I actually learned about photosynthesis just now.
Yeah, I got to Photoshop like a picture of a protein on Ryan Gosling's like shirtless body, and I was like, yeah, this is science communication.
Changing the world here in many ways.
But yeah, it was interesting because I had given pub talks before, but then after doing the science talk, like the science comedy talk, I had people coming up and asking me about carboxysomes. And I was like, oh, you really understood it. That's amazing. And like, you find it interesting. That's really cool.
Taylor, how did you even get into synthetic biology? How did you end up in this niche little part of the science world?
Yeah, I've always been into science, which is probably what everyone says, but it's also true.
I wasn't.
Really? Ah, cool. I actually almost majored in English.
Oh, see, no, I was horrible at English. I was really good at science, but I would say I was into it. I was just kind of like, it just comes to me. And then when I finished school, they were like, what are you going to study at university? And I'm like, I don't know. What's the hardest thing to get into? And they were like, science at Melbourne University. I was like, all right, put me down for that, but I don't think I'll do it. And then I just did it. And I love science now, but yeah, it took me a few decades to get there.
Okay, yeah, no, that's super fair. Yeah, I was into it when I was a kid, like catching bugs and watching the Weather Channel. I was like really into the Weather Channel when I was like 8 or 9.
I think everybody wants to be the weather person at one point.
Yeah, it's the coolest job. I'm still kind of upset that I didn't pursue that further. But when I was in high school, I got the opportunity to intern at a biotech company for 2 summers. They just had this like special program for high school students. And it was so much fun, and I really fell in love with proteins there because that was the first time I ever started working with proteins. And it just grabbed me in a way that I didn't really expect. And so I entered uni expecting to be pre-med because I also didn't really know if, as far as I knew, if you like science, you become a medical doctor. I didn't realize there were all of these other things that you could do instead. And then, I think a month into my first year of uni, I switched my major to biochemistry and was like, I want to get a PhD and I want to work on proteins. Because I sort of noticed, like, every time my lecturers talked about the human body, I was like, okay, cool. But every time they talked about proteins, I was like full alert, like so excited. And I was like, okay, I should probably lead into that.
Yes. Yeah, definitely. Good call. And then that just transitioned.
Yes. I just did biochemistry. I did some protein evolution research when I was in undergrad, and I thought that was super interesting. But I've always been really into protein structure. Because they're, like, if you see pictures of proteins, they're amazing. Like the intricate folds, like the little helices and the sheets.
So if you Google protein structures, it kind of looks like a child took a few different colored textures and just scribbled on a page. I mean, they start out simple in a straight line, and then they curl up like an extended pigtail, or these long folded sheets like napkins. And then they just go ham and fold in and around themselves, making these insane tangles of amino acids. I mean, I don't know how scientists can even begin to map out or understand these structures. But I guess that's why I'm not a structural biologist. Dr. Taylor is.
And so then my PhD was in structural biology, where I was solving the three-dimensional structures of proteins.
Right. Ooh, intense.
Yeah, it sounds intense. I guess it is intense, but it was really fun. Like, I really, really enjoyed that work because I was interested in protein structure, and so it was just a really, like, natural fit for me. And then after my PhD, I actually left research for a little while, and I ran this program out of the University of Technology, Sydney, called Steampunk Girls, which is an educational program for high school girls who are interested in STEM and integrating the arts and humanities with STEM. And so I got to work with schools all over New South Wales and got to see like these amazing projects that these girls were coming up with that sort of integrate art and science. Like one of them, the high school was really near like an assisted living facility. And so the girls would go over with like VR and liquid nitrogen ice cream and hang out with the residents there. And they formed these like beautiful relationships. Is like incredible to see. And it was just so like empathetically driven. It was amazing. And there are a million projects like that. And I was really, really fulfilled and like sort of saw the power of just thinking about science in a different way or talking about science in a different way. Because even now, like the work that I do is all empathically driven. Like all science, you're trying to make the world a better place. You're trying to understand things better to make the world a better place. And I feel like spending some time really like intentionally thinking about that was amazing and really set the stage for me to do something different in my postdoc and sort of think about things in a different way. And so then I saw the opportunity to get into protein engineering, which was something I was interested in, but I didn't know how to really step into it. And then when I joined the lab that I'm in now and postdocing in, I love protein engineering, and I really fell in love with synthetic biology because it's everything that I loved about my role running Steampunk Girls of like empathically driven science, and sort of having people and society at the core of that. So yeah, it felt like kind of a weird path and sort of got off for a little while, but everything has sort of informed the work I do now and the stuff I'm passionate about now.
Absolutely. When you said you got out of research, I thought that would be interesting. But doing the Steampunk Girls thing sounds incredible. I often come into this topic about diversity in STEM, and I think that's super important to get more people of all genders and ethnicities and abilities and whatever to get involved in science, engineering, technology, mathematics. Doesn't matter who you are. If you're curious or you think you can, you're empathetic to make the world a better place, like you said, then there's a place for you at the table. So it's great that you did that, and that actually brought you back into the world of biochem and synthetic biology. It's a cool little path. So I love that.
Thank you. Yeah.
What do you do when you're not doing science? What do you do when you're outside of the lab?
That's a great question. I do a lot of outreach, which I guess is technically outside of the lab, but still part of the whole science bit. So I've spent like this last year doing comedy. I also worked with a bunch of other people. So I'm part of the ARC Center of Excellence in Synthetic Biology, and we have an amazing outreach group. We're super passionate, excited people, and we made a card game called Remediate. And so the whole goal of the game is to try to engineer a microorganism that can remove the most plastic from a polluted environment. And so it actually puts players in like the driver's seat of doing synthetic biology, but with card games and with cute little characters and like fun mechanics that let you steal stuff from your friends.
This game actually looks epic. It's a card game where you get to create your own little microbes with different abilities and strengths and weaknesses. And the goal is to be the best at bioremediation, using your microbes to clean up the environment. I definitely want to get my hands on this game when it's released. And note that Taylor says it's coming out next year, but we actually spoke in December of 2023, so it should be released sometime in 2024. Check out the link in the show notes and get excited for Remediate.
And that was another like sort of combining science and art and society into this like neat little package that is not available yet, but hopefully will be next year.
This game, I'm going to link in the show notes below because you sent it to me and told me about it, and I was like, oh, that's cute. I feel guilty, but I wasn't so sure about it. But when I actually looked at it, it actually looks like such a fun game. Forget about Monopoly and Scrabble and anything else. I would hands down play this. It's almost like it. I guess it makes you feel like you're saving the planet. The concept is just incredible. Like the art is incredible.
It's amazing. And I do want to say the artwork was all done by scientists in the center. So we're talking just like outrageously talented people. Yeah, they didn't let me near it, which I think was a good idea because I am definitely not artistically inclined. But yeah, the artwork's amazing. I feel like people's expectations are extremely low when a group of scientists is like, we made a card game. They expect it to not be very fun, which, yeah, I definitely have gotten that surprised response. And so I feel like I always have to back it up by showing pictures of it, or the actual game would be like, no, it's like real and professionally done and really, really fun.
There's like a 30-second video on the website, and it's just like, oh, wow. No, yeah, I would definitely, I would pay a lot of money for that game.
Yeah, the trailer was also made by a scientist in the center on our outreach group. So it was all like homegrown. We did it ourselves, which also makes us even more proud of it. But yeah, so Remediate, we got a Science Week grant from Inspiring Australia. And so we did events in some major cities and a whole lot of regional hubs around Australia around Remediate. So a lot of it was like with school kids or with local industry. And I will say, I did some of these events in Orange, New South Wales, and we had like a local industry networking night, so people working in biotech in Orange, which is a lot of people. They have an amazing community. And we all came together for networking, and we decided to play Remediate, and I saw all of these connections that we had just formed just absolutely breaking down over the competitiveness of this particular game of Remediate. And I was like, I don't know if this was the right call.
Turning friends against each other.
We became friends, then quickly became enemies, and then ended as friends. So it came back. But no, it was super fun. That was a really neat experience, and I'm excited to move towards finishing it, which we're hoping to do early next year. I think we're getting close to our final version, which we're hoping to be able to put in the hands of more people. So yeah, definitely if you're interested in Remediate, get in contact so we actually get it done quickly next year.
Yeah, definitely. And I will have to keep in touch, and when it comes out, I will send everyone an update because people need to play this game. Do you have any book recommendations? Do you read much? Is there any books you would like to share?
I don't read a lot of science outside of work.
Any articles you'd like to share? Just kidding.
I did read a book during uni that I really, really enjoyed. It's called Napoleon's Buttons. It's like molecules that changed history.
Oh, cool.
It was really well written, and I was also at a point where I was in uni reading a lot of textbooks, and so reading this book was a nice difference. But it goes through different molecules and the effects that they've had and how they've changed the world. So like molecules found in like cloves and nutmeg that helped people sort of kill the mites that were causing the Black Plague. That was one of the things that it opened on.
Napoleon's Buttons, How 17 Molecules Changed History is a book by Penny LeCouture and Jay Burrison, and the name comes from one of these 17 molecules, tin, which was used to make the buttons that held the coats and trousers of Napoleon's army. But in the freezing cold weather, that tin actually crumbled, and the army's clothing basically fell off, forcing them to abandon their invasion. But this book also talks about the science behind silk, glucose, cellulose, even nutmeg, and how these substances have changed the course of history in medicine, in politics, the environment, and how they've had a huge impact on society as a whole.
I can't remember all of them, but it's a really nice mix of like informative science and really cool historical storytelling.
Do you have any daily rituals that you swear by? Anything that you do every day to help you get stuff done?
I've gotten into running in the last year.
Nice.
And so I've found that that's like a really nice way to clear my head and sort of I think about my work a lot while I'm running. Sometimes to take my mind off how much my legs hurt.
Yeah.
But it's also like a nice way to to like work through problems, or I plan presentations a lot actually. Like if I know I'm gonna have to present, then I'll think about like, okay, how do I want to structure this? What data do I want to show? And then next thing I know, the run's over. So I found that to be really helpful.
You're like, oh, I just did 5Ks. Rad.
Yeah, yeah, cool, nice.
Good. That's good. Exercise is important. It's good to get out of the lab and outreach and actually get some fresh air. And even if you're still doing science in your head at the time, it's probably a good way to do it.
And if I want to turn my brain off, I really like, I like crocheting.
Nice.
And crocheting a blanket is like the most meditative thing because you're just doing the same thing over and over again for a really long time. And so I can just completely clear my head and just like, it's amazing. Yeah, I love crocheting. Crocheting and running kind of do the same thing for me.
It's so wholesome. And then you get a blanket. I love that.
Yeah, and I've made a lot of toys for the office that we've got in a little box for like in case of emergency. If you get very stressed out, there's a lot of little buddies in here.
You probably make like those little microbe characters like from Remediate.
Yeah, actually, I've started sketching out patterns for them. So
Excellent. I've got a few listener questions for you, Taylor, if you've got a moment to answer those.
But before that, each week here on Travelling Science, we make a donation to a charity of the guest's choosing. And this week, Taylor has chosen to make a donation to Deadly Science, a foundation that provides science resources to remote schools in Australia that are often underfunded. Deadly Science is working to improve the quality of education in these schools, and a donation has been made to them on behalf of Dr. Taylor Szyszka. And if you'd like to make a donation as well, that would just be incredible. I can't recommend the Deadly Science Foundation enough, and there's a link to them in the show notes just below. But now, back to Travelling Science.
The first question is from Tambani. They're wondering, very general question, how do you engineer a protein? Basically, what does that involve?
Yeah. So proteins are encoded by DNA. And so the DNA will sort of give the cell instructions on how exactly to build the protein. Because proteins are actually just long chains of amino acids. And there aren't that many. There's about like 20 or so that are used commonly in proteins. And so by changing the DNA sequence, we can change the amino acid sequence. And because proteins fold based on that amino acid sequence in a really complicated way that we're kind of just starting to understand a little bit, then we can make those changes and then try to get different characteristics. So that's the basic idea. I spent a lot of time staring at structures and thinking about how to change them.
Insane. So you're doing genetic sort of stuff as well. You're like a geneticist, a biologist, a chemist, a physicist, an engineer. Oh my God. Very cool.
Yeah. Biochemistry and synthetic biology require a lot of dipping into other fields.
Yeah, and like cross collaboration, I'm sure. It's great. Cool. That makes sense. Grimly was wondering, and you sort of already covered this, but they're wondering why work on photosynthesis? Isn't that already pretty good? You said maybe it's not as good as we thought.
Yeah. It's not as optimal as it could be. So that's what, and people have been working on improving photosynthesis, at least since like the 1950s or 60s, or maybe a little bit later. I think it's been at least like 50 years. I keep forgetting that it's not 2000. I'm still stuck there mentally.
Yeah. Are we making progress? Or like 70 years and nothing yet?
Oh, yeah. No, there's, we're working on a really specific solution to the photosynthesis problem, but there are a lot of folks working on different ideas. So trying to improve that naughty enzyme I was talking about before directly. And yeah, lots of other ways inspired by nature and trying to find different ways to improve photosynthesis. I'm not a plant person, which I think is important to mention. So we are collaborating with plant scientists. Because I notice sometimes I start getting lots of questions about plants, and I'm like, oh, if you saw the state of my garden, you would not be asking me these questions.
Struggling to keep cactuses alive, you know?
Yeah, I killed my coriander in one day.
Oh no, that's okay. Nobody likes coriander. Well, I don't like coriander. Awesome. Well, that makes sense. We're working on photosynthesis. So there you go, Grimly. Last listener question is from Masonite, who is wondering, are there any risks with synthetic biology? It sounds like it could lead to the next pandemic. I don't know.
Yeah, so that is a question I get a lot. Just because of the nature that synthetic biology is talked about a lot, it can seem really like sci-fi and like we're doing all these wild things. But as we were talking about before, it takes a long time for anything to go from, gee, this sounds like a good idea, to actually happening. It's like many years, decades. And another thing I really like about synthetic biology is that in the same breath that we're talking about these amazing innovations, we're also talking about social responsibility. And so, like, even within our Center of Excellence, we have ethicists and philosophers and social scientists, so that when we have an idea for a technology, we do that proof of concept, we show we can produce it at scale, which again, a lot of time. Then it's sort of like, okay, how does this fit into society? Is this, and then sort of working through those regulations. Australia has incredible regulations for gene technology. They're incredibly effective. And so, yeah, so there's a whole lot of intermediate steps. So when I talk about improving photosynthesis, it's not going to happen tomorrow. We're still working in clear water in tubes. And so it's many stages down the line. And then you bring more people to the table when you get to those stages to make sure everything is going to be done properly.
Yes, you're not just doing these crazy experiments in the lab, which could turn into anything. I'm sure it's very regulated, and there's a lot of incredibly intelligent people like yourself making sure that everything is done to the best possible.
Yeah, and I'm definitely not an expert in like the regulations and science and society, and so that's why we have folks who are sort of along to help with these sorts of things. So yeah, everything is done with sort of that mindset of like, how does this fit into society, and sort of keeping that integrity and that responsibility in mind.
Fantastic. There you go, Masonite. Thank you so much, Taylor, for answering those listener questions. And just to finish off, I've got a lightning round, if you're ready.
Yeah, let's do it.
Yeah, alrighty. Pressure's on. Here we go. First question. As a protein engineer, do you drink as much as a normal engineer? Sorry, engineers.
I actually don't drink, so probably not. I don't know what the normal amount is, but I don't drink, so.
Right. Second question. If you could have done something completely different with your life, if you, say, never got into biochem and synthetic biology, what do you think you might have done?
I feel like I'd want to be a writer. I very nearly majored in English at uni. I've always loved reading. I've always loved writing and like coming up with creative ideas, and I use those skills in science. But yeah, I feel like I would have gone into something like that.
I would have liked to have read one of your books.
Maybe one day, who knows?
Yeah, totally. What do you love most about synthetic biology? What's your favorite part?
I love the possibilities. It's a young field, and it feels like there's no stupid ideas, at least in the beginning stages. Like when we go to conferences and stuff, we just have really fun conversations about how we could connect different ways we work. And so, yeah, I just love the possibility of it.
Nice. What do you hate most about synthetic biology? Is there any awful parts?
I mean, I guess I'll bring it back down to like maybe like day-to-day working in a lab. A lot of experiments don't work the first time. That's just the reality of doing science. But yeah, it can be sad, and that's actually something you have to learn how to deal with pretty early on, or else it'll get really crushing.
Totally. Yeah, lots of failures. If you had, say, a $100 million budget, something crazy, what type of research would you be investing that in? Where would you be focusing that work?
I'm really interested, like working on Remediate's made me really interested in bioremediation. So using microbes to clean up the environment, that's something that I find really cool. So I think I would want to really invest in sort of that kind of area.
I love that. Yeah, that's great. And final question, do you have a favorite protein?
Well, I mean, I feel like I kind of have to say encapsulin. But I'm also really interested in ice-binding proteins. I've never worked with one. I've just come across like reading interesting things. But yeah, so like Arctic fish will have these proteins because, you know, they're swimming, they need to not freeze. And so these proteins will actually like bind ice crystals and stop them from growing.
Wow, that is interesting.
Or like on the flip side, if you want to make ice or fake snow or stuff, you also have ice nucleating proteins, so proteins that can help ice crystals grow. Yeah, I just think they're super cool because they like you're kind of like rearranging like the water bonding networks. It's like kind of like water bending, but at like a protein level. It's really cool.
They sound like some really cool proteins. Taylor, thank you so much for coming on Traveling Science today and sharing your wisdom and your story, everything you're doing in the world of synthetic biology and with bioremediation, and all of your science outreach is just incredible. You really are helping to change the world. So thank you so much for everything you do. And just to finish off, considering everything we've discussed here today, I wonder if you've got one final piece of advice you'd like to share with our listeners, something maybe they could take home and apply to improve their lives in some way.
Yeah, absolutely. This is something that I've started to tell my students and stuff. So I'm actually publishing a few papers next week on projects that happened kind of by accident. We were trying to do one thing, but it worked a different way, and we completely went down that little tangent and came up with something really cool. So it's this idea of leave room to be surprised and be able to pivot. In something that you're doing, you might have I know I can be very narrow focus on an idea or something that I want, but this idea of leave room to be surprised. You never know what's going to happen, and interesting opportunities that might present a tangential path to somewhere else.
So there you have it, legends. That was Dr. Taylor Szyszka, protein engineer, synthetic biologist, superstar of STEM, stand-up scientist, and crochet queen. And if you want to learn more about Taylor's work, you can follow her on Twitter and LinkedIn at Taylor Szyszka. That's SZYSZKA, obviously. And check out her science comedy stand-up as well. It is fire. And those links are all in the show notes just down below. And I love Taylor's little bit of advice at the end. Leave room to be surprised. You never know what's going to happen, whether it's an experiment in the laboratory or something in your daily life. Sometimes things don't go as expected, but that's okay. Stay positive and leave room to be surprised, because sometimes the unexpected things that happen in our life can turn out to be amazing opportunities. Just be ready to pivot, to go with the flow. And that surprise that many people might consider to be negative could actually work out in your favor. So thank you, Taylor, for that little bit of wisdom. And thank you, legends, so much for listening this week. I truly appreciate you, and you're the reason I create this show. So make sure you're subscribed for more Traveling Science every single week, and follow Traveling Science on Instagram to see who we're interviewing next. And there you can also send in your own listener questions. Finally, if you're more visually oriented, feel free to check out our Traveling Scientist YouTube channel. And as always, if you stick around to the end of the episode, I always leave you with a little secret. And this week's secret is that I enjoy country music. There, I said it. Look, I'm not obsessed with it, but I don't hate it either. See, I listen to all genres of music. Rock, classic, jazz, rap, whatever. But I used to tell people that I will listen to anything except country. And then one day I said that to a Texan in Texas, and he was like, "Boy, you don't say stuff like that around here." Then he proceeded to drink beer and auditorily force feed me country music. And look, there was actually some decent stuff in there. And so ever since this southern musical revelation, I've found that, well, I love me some banjo. I sing Johnny Cash at karaoke. And when people ask me what music I like, I say that I'll listen to anything. Even country. Anyway, that's all for today, legends. Thank you so much for listening, and I'll catch you all next Science Sunday. Cheers.